everyone. Today we are going to resume the chapter Human Reproduction. We have already discussed about reproductive organs in that is male reproductive organ and female reproductive organ. Now today we are going to discuss main event, that is gametogenesis, that is formation and or production of gametes in humans. All of us know that there are two gametes which are produced in humans, that is sperms in males and ovum in females. Okay, and the production of sperms takes place in testis in the males, and production of ovum takes place in the ovaries in females. Both of these events are known as gametogenesis. Okay, so today we are going to discuss about the first one, that is spermatogenesis. Okay, now when we talk about spermatogenesis especially, it is the process of formation of sperms, as the word itself is describing itself. Spermato here means sperm, genesis means synthesis or production. So the production of germs, so the production of sperms in testis in the males is known as spermatogenesis. Okay, and this process starts from the spermatogonia. So spermatogonia is also known as male germ cell. Okay, and always remember that this process starts at puberty. Okay, so let's move on to the production of sperms, how it takes place, and what is the location where it takes place. Okay, now before moving on, it is very important to understand that um, when this process starts. Okay, so we will discuss it later on, but I'm giving you just uh, a little bit of information that initiation of spermatogenesis starts at the age of puberty in males, and it is only because of secretion of a hormone, that is GnRH. So I hope you can recall that GnRH is gonadotrophin releasing hormone, which is released from hypothalamus, and it further helps in initiating this process. And how it is initiated, we will discuss it later. Okay. Now, when we talk about spermatogenesis, we have discussed earlier that it takes place in testis, and the location in testis is seminiferous tubules, where spermatogenesis takes place. Here you can see an electron uh, picture, electron microscope picture, where you can see a sectional view of testis. Here you can clearly see that this is a seminiferous tubule. Okay, and we will further discuss about different types of cells or different levels of cells which are found as per the stage of maturation, which you will understand later. Okay, so this is just a sectional view of seminiferous tubules. Okay, now before moving on, it is very important to understand that how this process of spermatogenesis starts. Okay, now this process of spermatogenesis start in the testis. Okay, from a type of cells which are known as spermatogonia. Okay, for spermatogonia, this word is singular, and the plural for it is this word is plural. Spermatogonia is plural and spermatogonium is singular. Okay, so spermatogonia are those cells which are responsible for the formation of, for the further formation of sperms. Okay, how? So always remember that in the testis, spermatogonia are produced in large number, and they are produced through the process of mitotic division. Okay, now you know that in mitotic division the number of chromosome remains same. So here you can see that in spermatogonia, these cells keeps on multiplying inside the wall of seminiferous tubules through mitotic division, and the number keeps on increasing and they are diploid. That is why 46 chromosome is written here. 2n is 46. Okay, so the condition is diploid. Now what happens after some time? Some of these spermatogonia, they undergo first meiotic division, and those spermatogonia which undergo first meiotic division are known as primary spermatocyte. Okay, now these primary spermatocyte after first meiotic division, see, before first meiotic division they are diploid, but after first meiotic division they become haploid. Okay, and now they are known as secondary spermatocytes. Okay, so always remember that the diploid condition changes when they reach at the stage of secondary spermatocyte, where the number of chromosome is halved. Okay, this after second meiotic division, secondary spermatocytes are known as spermatids. Okay, so they are known as spermatids. Again, they are having 23 chromosome, that is a haploid condition. Okay, now spermatids are then further, you can say, matures, or they transforms into spermatozoa. Okay, spermatozoa, and they are also known as sperms. And this process of transformation of spermatids into spermatozoa is known as spermiogenesis. Okay, we call it spermiogenesis. And after their maturation, after spermiogenesis, the sperm heads, they remain embedded in the Sertoli cells. There are other type of cells found which are responsible for providing nutrition. So they remain embedded for certain period of time in Sertoli cells, and finally they are released from seminiferous tubules. And the release of sperms from seminiferous tubules now is known as spermiation. Okay, so there are two terms in it, always remember. First one is spermiogenesis, that is the transformation of spermatids into spermatozoa, and the release of sperm is known as spermiation. Okay, so these are certain important events. Okay, now let's move on. Now here you can see a sectional view, a diagrammatic representation of seminiferous tubule, which is very clear. Now here you can see that inside seminiferous tubules, okay, spermatogenesis is taking place, and here you can see that the cells near this epithelium are actually spermatogonia. So all of them are spermatogonia. Okay, now spermatogonia keeps on increasing with the help of mitotic division, I told you, and just after that comes another stage, that is of primary spermatocytes. So these are primary spermatocytes, some of the spermatogonia which are selected for first meiotic division. And just after that, first mitotic division, they forms secondary spermatocyte. After primary spermatocyte, they form secondary spermatocyte after completing their first meiotic division. So they are haploid. Okay, and these spermatocytes then later on, which are haploid in nature, transforms into or matures into spermatid. Okay, it transforms into spermatid. Spermatid undergoes further, you can say, they are uh, they divide further, or you can say that they transforms into, they don't divide but they transform, matures into sperm cells, or which are also known as spermatozoa. Okay, so this process of transformation of spermatid into sperm cell is known as spermiogenesis. Okay, so this diagram is clearly depicting different types of cells, where you can see spermatogonia near periphery, then comes primary spermatocyte, then comes secondary spermatocytes, and finally comes spermatid, which further transforms into sperms or spermatozoa, and later on they are released, and the process of release is known as spermiation. Now see, this is a schematic representation of spermatogenesis, which which will help you to understand and retain this how this process of spermatogenesis. Okay, here I'm repeating it once again. Let's revise it together. That in spermatogenesis, okay, mitotic division takes place in spermatogonia, which divides and redivides, and some of the spermatogonia are further, okay, they form primary spermatocytes. Okay, these primary spermatocytes will now undergo meiotic division, and that too the first meiotic division. After first meiotic division, each primary spermatocyte will divide into two, having haploid. Okay, they will divide into two, and they are known as secondary spermatocytes. Okay, and they will be haploid. Now, secondary spermatocytes will further undergo second meiotic division, and they will form four haploid cells. Okay, having half number of chromosome, that is 23 chromosomes. So primary spermatocyte will have 46 chromosome, whereas spermatids will have 23 chromosome. Okay, these spermatids now further undergoes the process of transformation, and they form sperms or mature sperms, or you can say spermatozoa, and it is this process is known as spermiogenesis. And they take nutrition from Sertoli cells, and finally after a certain period of time they are released from seminiferous tubules, and this process is called spermiation. Okay, I hope it is clear. Now comes the very important role of hormones. Okay, but before moving on, now this diagram is clear. I hope now you will understand this diagram, that on one side you can see they have shown testis. Here they have shown seminiferous tubules. If you will enlarge it, this will look like this, where you can see various stages of spermatogenesis. Okay, where you can see spermatogonium, and Sertoli cells are present inside it. Then again there are various types of cells which further mature, so differentiate finally into spermatozoa or sperms. Okay, and all this process takes place in testis, and that too in seminiferous tubules. Okay, now I'm going to discuss about the role of hormones, which is very important in this. Why? Because hormones are responsible for initiating the process of production of sperms, that is spermatogenesis in males. Okay, now as I told you earlier that to initiate this process, gonadotrophin releasing hormone GnRH. GN stands for gonadotrophin, R stands for releasing, H stands for hormone. So GnRH is released from hypothalamus, from the brain. Okay, and again it acts on the anterior region of pituitary gland. Okay, so it stimulates it to synthesize and secrete two hormones. One is called LH and the other one is called FSH. When we talk about LH, LH is known as luteinizing hormone. FSH, I hope you can recall, follicle stimulating hormone. So anterior pituitary gland secretes two very important hormones, LH and FSH. Okay, now LH directly acts on Leydig cells, which are the very important cells, and these Leydig cells are responsible for secretion and synthesis of androgen. Okay, in males, one of the androgen found is testosterone. So Leydig cells acts on um androgen. Okay, and they secrete androgen, that is testosterone, uh, and this helps in the process of spermatogenesis. Okay, so it stimulates, or you can say helps in the process of spermatogenesis. So how will you remember? Luteinizing hormone starts with L, and it will acts on another types of cells, Leydig cells. It also starts with L, so it is easy, LH for Leydig cells. Okay. On the other hand, FSH is follicle stimulating hormone, and it acts directly on Sertoli cells. Okay. Sertoli cells, and it stimulates a very important process, that is spermiogenesis. So when it acts on Sertoli cells, it helps in secretion of some factors which further help in the process of spermiogenesis. And spermiogenesis means release of sperms from seminiferous tubules. So these are the roles of different types of hormones. It is often asked in board exams also, that explain how significant is the release of or secretion of these hormones during the process of spermatogenesis. So you have to explain their functions in it. Okay, I hope it is clear. Now let's move on. When we talk about spermiogenesis, it means that spermatozoa or sperm is completely formed, and now it is released from the seminiferous tubule. So it is now very important to understand what exactly a sperm looks like, and what is its structure, and reason behind its structure also. Okay, now there is a very clear picture of sperm. You can see this is a ultra structure of sperm shown here, where you can see that the entire sperm is made up of three important parts, that is head, neck, and the last one is the tail region. Okay, this whole sperm is actually covered by, or you can say enveloped by, plasma membrane. And this before let's move on from the first part, that is head. Okay, so when we talk about head region, it is a very elongated, or you can say a compact structure, having having haploid nucleus in it. Okay, so it contains, the head region contains the haploid nucleus, um, uh, and the anterior region is covered with a cap-like structure. That is, this region is covered with a cap-like structure, and it is called acrosome. Okay, so acrosomes is very important because it contains certain enzymes, or you can say hydrolytic enzymes, and their function is very important during fertilization. When sperm uh, it fuses with the ovum, during that time it helps in acrosome, the enzymes present in acrosome helps in dissolving the membranes of the ovum, so that it can help in so it is actually indirectly helping in fertilization. Okay, so this anterior end is a cap-like structure which is called acrosome, having enzymes which helps in dissolving membranes of the ovum during fertilization, for fertilization. Okay, then comes neck region. Okay, this neck region is again very important. This neck region contains centriole, two centriole, and we can say that proximal centriole is there, and then there is distal centriole is there. Okay, so one centriole which is present in the neck region is called proximal centriole, and the another one is present in tail region, so that is called distal centriole. Proximal, you know the meaning also, you can remember it like this: proximal centriole is close to head, distal centriole is away from the head, it is present in the tail region. So it is believed that proximal centriole is important for the first division or cleavage of zygote. Okay, and distal centriole is connected to the tail filament. Okay, now comes the middle piece. Okay, now this is the middle piece. Okay, this middle piece contains number of mitochondria. You know that male gametes cannot move on their own, so they need some energy for the movement, so that energy for the motility comes from number of mitochondria present in this middle piece. Okay, and the last one, that is how it is moving because of its tail. Okay, the sperm can move into the female reproductive tract during fertilization, for the fertilization, with the help of tail. So it its movement is like flagellum. Okay, I hope it is clear now. Now here also you can study, you can see that sperm is divided into head, middle piece and tail. Head contains a haploid nucleus. Anterior part is covered by acrosome. Then middle piece contains mitochondria, and tail helps in the motility of sperm, of movement of sperm, so that it can reach at the site of ovum. Okay, I hope it is clear. Now moving on to the next important process. We have already discussed about spermatogenesis. Now moving on to oogenesis. Okay, now I want to add one more information before moving on, that um, the you must have heard about there are terms like we have discussed about epididymis, vas deferens, seminal vesicle, all of them along with the sperm. Okay, the secretions from all these accessory ducts as well as sperms, or you can say together with sperm, collectively forms semen. Okay, so this is again important for exams. It can be asked that what forms the semen. Okay, now moving on to the next part, that is production of female gamete, and the production of female gamete is known as oogenesis. Okay, now oogenesis is a very important process, and the most important point is that oogenesis is initiated, or you can say begins, at embryonic development stage. This is very important. We discussed that spermatogenesis starts at you can say puberty, whereas you have to remember that this starts at embryonic stage. Okay, and in embryonic stage, gamete mother cells, millions of gamete mother cells are formed, and these millions of gamete mother cells are known as oogonia. Okay, and so you can say that in each fetal ovary during embryonic stage only, millions of gametes are already formed. Okay, and so no more oogonia will be added after birth. Okay, before birth, oogonia are already formed in number of millions, and they will not form further after birth. Okay, so their oogonial cells enters into meiosis 1, and whichever oogonial cells are formed, see one more thing which is very important, and you can understand it very easily, that like spermatogonia are formed by mitosis, here also oogonia are formed by mitosis in large number, and after some time some of them will undergo, most of them, or you can just most of them will undergo meiosis 1. Okay, and now they are called as you can say they are called as those which will undergo meiosis 1, they are called as primary oocyte. Okay, but here the difference is that meiosis 1 is not completed. In primary oocytes, the cell division remains suspended, it stops in prophase 1. Okay, so I'm repeating it once again. Oogonial cells enter meiosis, okay, enter meiosis 1, and then the primary oocyte remains in prophase 1, okay, in the arrested state, or you can say that meiosis 1 stops at prophase 1. Okay, I hope it is clear. Um, Now see, there are certain changes which takes place in it. These changes are that those primary oocyte which are at arrested state, they will now undergo uh, further changes. Not the cell division, but some other changes will take place in primary oocyte. Okay, so this primary oocyte, which is at first prophase stage and it is arrested, it means suspended at this stage. No, it is meiosis 1 is not going on. It has already stopped. So there are certain changes which takes place during primary oocyte, which is called where follicles are formed. Okay, and this primary oocyte, okay, they undergo this primary oocyte, it becomes surrounded by a layer of granulosa cells. Okay, and this is known as primary follicle. And after a certain period of time, primary follicles becomes surrounded by more granulosa cells. So an external layer is also formed and it is called secondary follicle now. Okay, I'm repeating it once again, that primary oocyte becomes surrounded by granulosa cells and it is called primary follicle. They are further surrounded by more granulosa cells and there it is known as secondary follicle. Okay, now there is one more thing which you have to understand, or that is large number of follicles. Out of these follicles which are formed, which are in millions, okay, um, many follicles degenerate during this stage. So although the number was millions, but now it will be around 60 to 80,000 which will be left in ovary. Okay, 60 to 80,000 only. And this degeneration of follicles before puberty, it takes place before puberty. The degeneration of large number of follicles, which reduces their numbers from millions to 60,000 to 80,000 before puberty, is known as follicular atresia. Okay, and so after puberty only 60 to 80,000 follicles are left in ovary. So they are further surrounded by granulosa cells. These primary follicles are now further surrounded by granulosa cells and they form secondary follicles. Okay, now when they form secondary follicle, after a certain period of time, the layer of cells divides into, okay, two layer, external layer and internal layer. So the external layer is known as externa and internal is known as interna. Okay, now this is known as tertiary follicle. When this differentiation of layer starts, then it is known as tertiary follicle. Then this tertiary follicle uh, continues its development like this, and it finally completes meiosis 1, and it contains, you can say that it contains a fluid filled cavity. Tertiary follicle can be recognized by having a fluid filled cavity, and this is called as antrum. Okay, and it has got outer layer which is called externa and internal layer called as um, interna. Okay, so this is the, and as soon as tertiary follicle is completely formed, first meiotic division also completes with it. Okay, now when we talk about this, as soon as the first meiotic division is complete, you will see that primary oocyte will form secondary oocyte and first a small structure. So this division is unequal. A small structure is formed and a bigger structure is formed. Small one is called as first polar body and second one is known as secondary oocyte. Okay, now this is believed that first polar body has no function, so it degenerates itself after some time, and secondary oocyte now further divides through second meiotic division and forms gives rise to another unequal division where a small structure is formed and bigger structure is formed. Small structure is known as second polar body and bigger one is known as, you can say, ovum, which is haploid. Okay, now although it is shown in the structure, but I would like to add to it. Okay, that the secondary oocytes starts at second meiotic division, but it remains in a suspended state, which is metaphase 2. Okay, and it is observed that it completes its second meiotic division only when a sperm enters into it. Okay, and finally it completes its second meiotic division, but we will discuss it later on in a in a more explanatory way. I will describe it more because it will get little confusing. Right now, just understand this, that oogonia, okay, oogonia are formed, okay, through mitotic division, and they are in millions, okay, and during fetal stage, and uh, then they come into, or they divide further and form primary oocyte. That is the first meiosis, but first meiosis is not completed at this stage and they remain suspended at prophase 1. Okay, prophase 1, and this is primary oocyte. And now this primary oocyte undergoes various changes. Okay, these various changes are, it is surrounded by very number of layers of granulosa cells, and granulosa cells, as per advancement, like it forms primary follicle first, secondary follicle, and finally tertiary follicle. And as soon as it forms tertiary follicle having a fluid filled cavity, it completes its first meiotic division. Okay, and it forms a secondary oocyte and a small first polar body. And after completion of second meiotic division, it forms second polar body and ovum, okay, which is haploid. And here it is written as ootid also. So at first polar body and second first polar body and second polar body has no functions, so it is believed that it degenerates after some time. Okay, so this is about oogenesis. I also discussed about follicular atresia. Always remember that before puberty, those millions of oogonia, some of them degenerates. Okay, millions of these you can say primary oocytes. Out of these millions of primary oocytes, some of them degenerates, and this is known as follicular atresia. Okay, and therefore before puberty only 60 to 80,000 primary oocytes are left in the ovary for further development. Okay, so here you can see another structure. This is a diagrammatic, or you can sectional view of ovary, where you can clearly see that primary oocyte is changing into primary follicle. Okay, so they have shown primary, secondary, or you can say this is the tertiary stage, where you can see fluid filled cavity and nutrient rich cytoplasm is filled inside it. And after completion of it, secondary oocyte is formed. Okay, and secondary oocyte is formed along with polar body. Okay, now the secondary oocyte further divides to form a second polar body as well as ovum. Okay, so later on we will discuss about these structures also. Um, these structures also. Um, it is believed that when it completes its formation, okay, the when ovum is released, the entire structure, that is when ovum is released from the secondary oocyte, you can say that the remaining structure is known as it is earlier known as Graafian follicle. The tertiary follicle is it changes further, it matures and forms Graafian follicle. And from Graafian follicle, when ovum is released, it is known as ovulation. Okay, and the structure which is left after release of ovum is known as corpus luteum. Okay, we will discuss later on that what is its function and so on. We will discuss it later. Okay, I hope it is clear now. Always remember that it is in board exam, it is asked what is the basic difference between spermatogenesis and oogenesis. Okay, they also ask the stages of formation of male gamete, that is spermatogenesis, as well as female gamete. Okay, this is another structure, or you can say sectional view of ovary, okay, where they have shown various types of or you can say various stages of this oogenesis, okay. And there is another stage which they have shown, which is the form, this is a mature ovum, okay. And when we talk about this structure, you can see that the secondary oocyte forms a new membrane during that development only. The outer layer is known as zona pellucida, okay. Or you can say secondary oocyte is, you can say that it is surrounded by a membrane which is called zona pellucida, okay. And there is outer loose cells, layer of loose cells, which is called corona radiata. So you can understand it like this: ovum is covered by a membrane which is known as zona pellucida, okay. And the outer cells in secondary oocyte are known as corona radiata, okay. So this is another structure, and other stages are similar in it. This is a sectional view of structure of ovary, showing various stages in it, okay. There again, if you want to recall it by that how primary oocyte changes into what changes it undergoes, you can see this flow chart where you can clearly see that the primary oocyte formed, okay, it is surrounded by layer of granulosa cells, okay. Now the primary oocyte is in arrested stage of first meiosis, and this primary oocyte then it is covered by a layer of granulosa cells known as primary follicle. It is surrounded by more layers of granulosa, so it later on forms secondary follicle. And after some time, it develops further, where it contains outer layer which is called externa and internal layer called interna, okay. And it is characterized by a presence of fluid filled cavity called as antrum. So this is known as tertiary follicle, okay. And as soon as tertiary follicle is formed, it completes its first meiotic division to form a haploid secondary oocyte, okay. And this tertiary follicle then matures to form another structure that is called as Graafian follicle. So now I hope this confusion is clear, that primary oocyte further develops into primary follicle, secondary follicle, tertiary follicle, and finally into Graafian follicle, okay. And from Graafian follicle, after some time, the secondary oocyte present in it forms a new membrane which is called zona pellucida, and it has a loose cover of cells which is called as corona radiata. And after some time, ovulation takes place. Ovulation, all of you know that it is a release of ovum from the ovary, is known as ovulation, okay. Now this is another chart. If you are having any confusions, you can see this chart also, where it is explained, various stages are explained in it, in the same manner the way I have explained, okay. And we will discuss later on about the role of hormones also. So if you want to understand and recall it again, then you can just go through this chart, okay. Now this is a sectional view of ovary, okay. This is a sectional view of ovum, sorry, where you can see the structure also. Now here you can see they have shown various layers, okay, in this. Now this is this structure where they have shown that how this ovum is released from the ovary. So various stages are shown here. It is also shown how this whole structure is ovary. This is egg. This is follicle. This is not very clear, but you can understand by looking at it. So I hope that you must have understood the process of oogenesis, and always understand it, make diagrams so that you can remember it, and try to learn it in steps so that whenever questions are asked, you can write it in steps properly and you can recall it by understanding it. You cannot cram this whole procedure, you have to remember it and understand it, and then you have to practice it in the same manner. Thank you.